This topic deals with protein synthesis. Almost 80% of our body, that means the dry mass, is made up of proteins. Proteins are large polymers, and they are made up of monomer units called amino acids. If these blue little circles represent amino acids, this could be amino acid 1. This could be amino acid 5. This could be amino acid 17. This could be amino acid 11. So, a series of amino acids joined, one to the other would then form a protein. In order to call the compound, or molecule, a protein, you must have more than 50 amino acids. From earlier knowledge, you will understand that enzymes and hormones are protein in nature. Protein synthesis occurs at the ribosome, a structure found in the cytosol of cells. Let us recap. Structure of the nucleus. On the outside is the nuclear membrane. Some books would refer to it as a nuclear envelope. We might find a number of strands, either collectively or singularly spread out in the nucleus, and this would then refer to your chromatin, or individually as chromosomes. This little dot indicates a hole in the membrane, so that will be your nuclear pore. And the denser structure within the nucleus would then be your nucleolus. Everything inside the nucleus that is indicated in blue would then be nucleoplasm. To understand protein synthesis, we need to break the process up into two stages. The first part is transcription, and it takes place in the nucleus, the second part being translation, which takes place in the cytoplasm. Firstly, transcription. During this process, the DNA, which was a double helix, unwinds. Very similar to that in replication, and then it unzips. Now, in DNA replication, both strands were copied, but in protein synthesis transcription, only a single strand is copied, and we form messenger RNA. So, this is messenger RNA. It was formed from copying one strand of DNA. In order to form this mRNA, free nucleotides from the nucleotide pool was used to form the messenger RNA. Messenger RNA then leaves via the nuclear pore. When it leaves the nucleus, it goes to the cytoplasm. The second stage in protein synthesis is called translation. This is obviously going to take place outside the nucleus, and it's going to occur at the ribosome, the site of protein synthesis. At the ribosome, the messenger RNA straddles itself around the ribosome. In order to join the ribosome, it uses rRNA, ribosomal RNA. The next step is that transfer RNA, symbolized tRNA, aligns itself along messenger RNA. It does so by using the three bases along the messenger RNA that we call codons. So, each three bases that are consecutive constitute a codon, and they're actually code for a particular amino acid. Each transfer RNA has a triplet code, and this triplet code is referred to as the anticodon. So, on the messenger RNA, you have codons. And on the transfer RNAs, you have anticodons. Once the transfer RNA positions itself on the messenger RNA, the amino acids are then sequenced. The amino acids then join to each other by peptide bonds to form your protein. The following is part of an examination paper, just to practice what we have learned. If I consider this diagram, it says, the diagram below shows a part of the process of protein synthesis. When I'm studying this diagram, I need to identify firstly, ribosome. Molecule X, single strand, molecule Y, I see letters all over. The question asks, name the stage of protein synthesis, represented in the diagram. To answer this question obviously, I can see ribosomes, so the answer to that is, cytoplasm. So if it's cytoplasm, then translation. The next question asks you to identify molecule X. So, molecule X, like I identified earlier on, was just a single strand, so it could only be mRNA. Molecule Y, if I look at it closely, shows 3 bases on one end, and amino acid at the other end, so molecule Y would then be tRNA. And then, the last question says, identify structure 1. Now structure 1 is talking about those triplet bases that are present on molecule X. So structure 1, molecule X, let's think again. Molecule X was messenger RNA, so the 3-letter bases on molecular RNA is codon. Well done. That would show the type of questions on protein synthesis.